Okay, so today we're going to look at one of our first case studies here, and that is how do genes respond to the environment? We know that there are cases where genes where genes get turned on or turned off depending on sensing what's going on around them, but how do they actually do this? And so bacteria can grow on different sources of sugars. And so just like we eat different foods, the bacteria need different kinds of sugars, but they also need different enzymes to be able to metabolize or break down those different sugars. But it's very wasteful for the bacteria to express and always have those enzymes around if that sugar's not around. So the bacterial genes are able to respond to which sugars are around. And so we can see in this graph here, people have found that in E. coli, that there was an enzyme called beta-galactosidase. ACE means it's a protein, and it basically breaks down the beta-gal linkage of the sugar lactose. And at this graph, we are just looking at the amount so the y-axis here is showing us how much of that protein is there, and that once there is not really any protein around, and then lactose is added, and then we get a large increase in the amount of beta-galactosidase, and then lactose is removed and the transcription stops. So now we have a really nice system or experimental setup to be able to ask, let me just go back to that slide, to be able to ask, when is lactose turned around? How is it that lactose turns on and makes the productions of these proteins? And so there were some scientists that realized that there was a special sequence of DNA in the bacteria that encoded genes that were really important, or had different functional segments that were important for this response to turning on lactose. So there was two proteins, lac beta and the lac P, which is the permease. It is the thing that actually transports lactose into the cell. And so they were already known to be proteins important for being around when lactose was there. But what about these other two segments? They knew that there was two other segments called lac I and lac O that were also important for the metabolizing lactose. But what do they actually do, and what's their function? So, two scientists, Jacob and Monod, decided to investigate this, and they made mutants in lac I and lac O to see the effect on beta-galactosidase or lac beta production. And now let's take a look at what they found. So they had different genotypes, and then they looked at the amount of beta-gal in minus lactose conditions and in plus lactose conditions. And so let's take a look and see what we have there. So we're going to start with, just as we always do, what happens under the wild type condition. We need that control to establish the baseline. So we have I plus, O plus, beta plus, and P plus. The plus here just denotes that is the wild type, the normal allele is what we call it. And remember, an allele is just different versions of the same gene. And so when there's no lactose around, they don't make beta-gal, just as we saw in that last graph. And when there is lactose around, we see lots of beta-gal. Then they asked, okay, they had a mutant. What happens if we mutate or basically break the lac I version? How does that change how beta-gal responds to lactose? And they got this interesting result that now the beta-gal protein is always produced. That gene is always on, no matter whether or not lactose is there or not. So it's not responding to lactose, it's saying, I'm just going to make this all the time. Then they had another version or another allele of the lac repressor. And so it's got a capital D there. And they wanted to say, okay, what does it look like? How much beta-gal is produced when that version is around? And they found this really interesting thing where this time now it was basically the opposite. So that no matter, even if lactose is around, it's still not making any of the beta-gal. And so their question was can this allele, this Lac ID, prevent beta gal accumulation even when even on a different DNA segment when it's elsewhere? And so they actually were able to make a diploid or put in another locus. They made basically a plasmid with the same genes and asked if that allele could now function on it. So just like we wanted the wild type version in the when we are haploid or there's only one allele around up here, we are also going to do see what our baseline is for when everything's normal, everything's wild type, but in two versions. So that's what this alleles are going to show. And so we can see that we have all the wild types and all the wild types, and it responds to lactose just as before, but this time we get a bit more beta-galactosidase accumulation, and that's not too surprising since now we have two copies of the beta-galactosidase gene. So then they mutated one of the two alleles into that dominant version of the gene. And so here is the dominant Lac repressor, but then we still have one version of the wild type copy around. And what did they find? This time it looks just like when there's one version around. This capital D version of Lac I is preventing the transcription of beta-galactosidase even on a different DNA locus. So we're still not getting it to respond when there's any lactose around. And so this suggested that it makes a protein that can diffuse and work on a different DNA segment. So now that they had a handle on what the protein, on what the Lac D was, the next question was, what about that Lac O? And so the Lac O, they were able to make a version where they broke it just like they did with the Lac I. So we can see this allele version here. And like the Lac D version, when there was no lactose around, still there's no beta-galactosidase made, but beta-galactosidase was also now not being made even when there was lactose around. And so again, they wanted to look and see, can this prevent accumulation of beta-galactosidase at a different loci? So they made the diploid again, and so now we have a diploid, and the Lac O only works in one copy. So let's, if we look here, we're still getting now beta-galactose response with in response to lactose being around, but it's acting just as if there was one copy. And so our Lac O, unlike the Lac I, can only work on the DNA segment that it's in, so this DNA segment here, and not on the other copy of DNA. And so that leads us to our next model, and we take a look back at our model. And so here is that DNA segment. We had Lac I, Lac O, and then Lac beta, and Lac P, Lac permease. And so the Lac I makes a protein that then represses the operator or the promoter that activates transcription of Lac beta and Lac P. And so when there's no lactose around, it sits on the promoter and makes it so that it cannot be transcribed. But when lactose is around, then we have lactose around, it actually binds to the Lac repressor, it leaves the promoter, and now transcription can happen. So through these experiments, it was one of the first demonstration of a gene set responding to its environment, and that was dependent both on a protein as well as on the small segment of DNA called the promoter. And the next follow-up case studies will really take a look at how does a promoter function.